I'm back. So in our last screencast, I got a little cut off. We were talking about how in transcription, DNA is used to generate messenger RNA, and we looked at the process of how RNA polymerase carries that process out. And then we talked about how that messenger RNA, it needs to be processed a little bit in eukaryotic cells at least, in order to get it ready for translation. And so in my analogy, if messenger RNA is a photocopy, processing is kind of like three-hole punching, adding a staple, and just kind of tightening things up so that the next phase works as well as possible. So the ends are modified. We add this little cap on the 5' end. We add this poly A tail on the 3' end. And then we have to do a little bit more work. We have to splice out these non-coding regions, which it may have surprised you to discover existed. So yes, in eukaryotic cells, there's these little weird interrupting sequences called introns. And so in splicing, we're getting rid of those introns, so only the coding sections are there. And this process is carried out by this little thing called a spliceosome. Now, an obvious question would be, well, why do these introns exist? Well, one benefit to having them is that it allows the same DNA to give rise to different types of messenger RNA. So if we transcribe this DNA, we get this RNA, but then by splicing it in different ways, we can get different types of messenger RNAs that are finally finished. And so we could splice out, for example, if we splice out exon 3, then we get this protein, which is missing this little region and is therefore slightly different. If we splice out exon number 4, we get a slightly different protein. It turns out this is much more important than we ever recognized for a long time. So before actually sequencing the genome, scientists expected way more genes than there are. The estimates were between 80 to 150,000 genes. Once they sequenced the DNA, there was this big puzzle when they realized, wait, there's only about 21,000 genes. How is that possible? Well, the partial answer is that many RNA molecules exist than do genes. So basically 80% of genes are alternatively spliced. So of these 21,000 genes, we get a lot of different types of messenger RNA and lots of different types of proteins. And so that kind of helps reconcile that difference between what we thought should happen and what actually happened, this alternate splicing. Introns also increase the probability of crossing over, because it increases the distance between coding portions of the genes. And remember, crossing over is where you get new combinations of genes, which contributes to genetic diversity. And so that could kind of explain possibly how these would have evolved to exist as well. And then finally, just a reminder, in prokaryotes, there is no messenger RNA processing. And that's because since there's no nucleus, there's nothing separating transcription and translation. So as soon as transcription generates that messenger RNA, translation immediately kicks in and starts to occur. So there's no processing in prokaryotic cells. Okay, sorry it got cut off there. This last part then is on translation. Translation is the process of producing the protein from messenger RNA. In other words, it's kind of like taking the recipe and making our pie. So where is this going to occur? Well, this is going to occur in the ribosome, which could be located in either the cytoplasm or the rough endoplasmic reticulum. So it's worth reviewing a couple things. So take a moment and reflect on these questions, hit pause, and then we'll talk about the answers. So hopefully you thought about these a little bit. So what are the building blocks of proteins, amino acids? How many are there? 20, and we talked about that quite a bit in the previous screencast. Does the order of the building blocks that make up a protein matter? Absolutely yes. That order is the primary structure, and that specific order causes it to fold in a very specific way to give rise to a very specific three-dimensional shape. And again, it kind of looks like that in real life, but we represent it like this to kind of help our brains wrap around it. What type of chemical reaction connects the two amino acids? It is a dehydration synthesis reaction, and then we form this what's called peptide bond, which is that bond between the two amino acids. So with translation, I really recommend that you watch this animation. It's the same one that you started with transcription, but now you're going to start at a minute 35 and go to the end because, I mean, just looking at this, it gets really complicated. Well worth pausing to watch that animation. Okay, players. Player number one is the processed mRNA strand from transcription. Obviously, in prokaryotic cells, it's just the mRNA strand because there is no processing. And so remember that this processed mRNA strand is broken down into codons, these 3 base units, which are each going to specify a particular amino acid based on the genetic code. And so we've already kind of talked about this, but GCU, take a moment. What would GCU code for? And if you find it on there, hopefully you said alanine. What does ACG code for? Find ACG. Do you see ACG? Threonine. And so you could go through and just kind of confirm that you know how to use this genetic code. So basically these codons are going to code for this specific order of amino acids. And note that UAG at the end is stop. So that's where it all ends. The second player is new to us, tRNA or transfer RNA. Briefly mentioned previously. This is key to the process of translation. This essentially translates the language of nucleic acids, which is the bases, into the language of proteins, which is amino acids. What does that mean? Well, if you look at this molecule, it looks like this. It's a type of RNA. But on one end, it has what's called an amino acid attachment site. So a specific amino acid is going to attach to this end. So tRNA kind of speaks the language of protein because it can attach to a specific amino acid. On the other end of the molecule is something called an anticodon. And the anticodon are 3 nucleotides that are complementary to the mRNA codon. In other words, this AAG is going to interact with UUC on the mRNA. So it also speaks the language of messenger RNA. I like to think of this, it's kind of like the Chinese English dictionary. It speaks both languages. It speaks the language of protein and it speaks the language of nucleic acid because different parts of the molecule interact with each part. Okay, and note that the amino acid is specific based on what that anticodon is. Okay. The next player is the ribosome. We've kind of represented these as dots so far, but now we're going to take a little closer look. The ribosome consists of both large and small subunits. And then there's these little kind of pockets or grooves in the ribosomes where tRNA molecules kind of fit in. They're labeled E, P, and A for Environmental Protection Agency. And also for this, A is the what's called amino acyl site. Just think amino acid. This is going to bind to the tRNA containing the newest amino acid. The P site, think peptide, that's going to hold the growing polypeptide chain. And then the E is the exit site. That's where the tRNA is going to exit. And again, these are located either in the cytoplasm or the rough ER. So how does this all go down? Again, that animation, I think, really helps. Here's kind of the big picture process. Here it is step by step. So just kind of zoom in on these pictures one by one. So first is what's called initiation. What happens is the ribosome, the messenger RNA right here, the tRNA, which is going to contain methionine, because remember, methionine is always the first amino acid. They're going to assemble. Okay, so this start codon is AUG on the mRNA. The anticodon UAC is going to ensure that this particular tRNA molecule containing this particular amino acid is going to interact right there. So the mRNA, the tRNA, the ribosome all kind of assemble into this structure, okay? And we're positioned. Our start codon has our methionine, our first amino acid. Then what happens is elongation occurs. There's a 3-base codon inside this A site now. And so the new tRNA with the appropriate amino acid based on its anticodon that will fit and match complementary to this will come in. Okay, so now that new amino acid is brought in, again, based on that 3-base codon. Then the polypeptide chain is going to grow. So this polypeptide chain from the P site, in our example, it just contained methionine so far, but in this picture it has several more. But that's just going to be transferred to the A site. So now this new amino acid is going to be connected to all the rest of that growing polypeptide. Then what happens is, oh, and that's going to form that peptide bond that we talked about before. Then what happens is the ribosome shifts position. So the amino acid that initially began in the P, or the tRNA that initially began in the P site, you can see that tRNA is now empty because it just transferred its polypeptide to this new amino acid. So the ribosome is going to shift, and now this tRNA molecule is going to find itself in the E site. This tRNA with the growing polypeptide chain is going to find itself in the P site, and the A site is going to be empty. Okay, so this whole ribosome just kind of shifted over to the right one, and that put this in the E site and this in the P site and created an empty site for A. Hopefully you can visualize that. So now the tRNA in the E site is just going to exit, and it can be recharged with the appropriate amino acid and recycled. And then we're kind of back to where we started again. Now the next amino acid comes in, kakunk. The growing polypeptide chain is added to that, kakunk. It shifts, this tRNA exits, and now we're ready to go it again. And it just keeps going and going and going. And again, that animation really helps. So we repeat for all the codons. And at the end, termination occurs. At the stop codon, a protein, not a tRNA, but a protein called a release factor is going to attach. And now, when we try to transfer that polypeptide to the next amino acid, there is no more amino acid there because it's just this weird protein. And so what happens is the protein tries to get transferred, but there's nothing to transfer to, and so it just falls out. And then once it falls out, that kind of causes the ribosome, the messenger RNA, and everything else just kind of disassemble, and we're done. That polypeptide is now generated and produced. So again, this animation, I cannot emphasize enough how helpful that is, but you can kind of see this process and how it takes place with this flow. Okay, so to recap what we have done, we started protein synthesis with the DNA. We take that DNA in the process of transcription. RNA polymerase makes our disposable copy of a segment of that DNA. This is our recipe. RNA processing takes place. We add the poly A tail, we add the guanine cap, and we splice out those non-coding introns. The messenger RNA then travels into the cytoplasm or to the rough ER, where it is joined to a ribosome, where translation is going to happen and build the protein or make the cake. And that, my friends, is the end of protein synthesis, a very complicated process. So in our next screencast, we will look at what happens to these protein products and then talk about the role of mutation in all of this.